Hello, welcome to the ScienceSloth Channel. Today we will be reviewing a paper from Cell called Psychedelics Promote Structural and Functional Neuroplasticity. This paper by Lai et al. Sought to document the effects of three psychedelics, LSD, DMT, and DOI, on prefrontal cortex neurons both in vitro and in vivo. The authors introduced the term psychoplastogen to classify these compounds that are capable of rapidly promoting induced plasticity. Researchers sought to elucidate how serotonergic psychedelics produce rapid and long-lasting antidepressant effects at the cellular level. Atrophy of prefrontal cortex neurons is thought to play a key role in depression and related disorders. However, such atrophying can be counteracted with compounds that promote plasticity in the prefrontal cortex, like these psychoplastogens. The first round of experiments involved treating rodent-cultured cortical neurons with DOI, LSD, and DMT to measure any resulting changes in morphological features via shoal analysis. Results showed all three compounds promoted neuritogenesis. In vivo effects of these compounds were tested in Drosophila larvae during the first and third instar with LSD and DOI only. Both increased dendritic branches of class I sensory neurons during both instars, with effects most pronounced during the first instar. Similar results in rodent and Drosophila models indicate that these psychedelics work on an evolutionarily conserved mechanism. In the second round of experiments, researchers treated mature rat cortical cultures for 24 hours with DOI, DMT, and LSD. They measured the number of dendritic spines with super-resolution structured illumination microscopy. All three compounds increased the number of dendritic spines. The new dendrites favor thin, immature spine types over more mature, mushroom-shaped spine types. Researchers then administered a 10 mg per kilogram intraperitoneal dose of DMT into the prefrontal cortex of adult rats and measured spinogenesis with Golgi-Cox staining. 24 hours after dosing, there was a significant increase in the density of dendritic spines on cortical pyramidal neurons, which was comparable to the same dosage of ketamine. This increase in dendritic spine density was accompanied by an increase in the frequency and amplitude of spontaneous excitatory postsynaptic currents. The next round of experiments sought to compare the effects of brain-derived neurotropic factor and DOI. Cortical neurons were treated with BDNF, DOI, and a combination of the two in order to see if they produce an additive effect. Shoal analysis revealed that BDNF at 50 nanograms per milliliter increased neuritogenesis to a similar degree as DOI at 10 micromolar, but no additive effect was produced when cells were treated with both. This indicated that they both worked through a similar mechanism. When cortical cultures were treated with ANA12, a selective antagonist of BDNF's high-affinity receptor, TRKB, neither psychedelics nor BDNF were able to stimulate neuritogenesis or spinogenesis, suggesting that these compounds promote plasticity through TRKB. Since TRKB is known to promote signaling through mTOR, researchers treated cultures with LSD, DOI, or DMT and rapamycin, an mTOR inhibitor. This completely blocks psychedelic-induced neuritogenesis, indicating that mTOR also mediates the plasticity-promoting effects of these psychedelics. Researchers concluded their paper with an investigation into whether or not the serotonin receptor 5-HT2A played any role in the plasticity-promoting effects of these psychedelics, due to this receptor playing a key role in the hallucinogenic effects of psychedelics. Rat cortical neurons treated with either DMT, LSD, or DOI were also exposed to the selective 5-HT2A receptor antagonist ketosarin. This completely nullified the neuritogenesis and spinogenesis effects previously elicited by the psychoplastogens. This suggests that each of these compounds are working through the 5-HT2A receptor. Through both in vitro and in vivo testing of various psychoplastogens, Lai et al. Were able to conclude that DOI, DMT, and LSD increase neuritogenesis, spinogenesis, and synaptogenesis, and this increase in plasticity is mediated by TRKB, mTOR, and 5-HT2A dependent mechanisms. Thanks for watching. The full paper will be linked in the description box below for those of you interested in reading it. See you next time!